Hello and welcome to the DDW Sitting Down With podcast series, where we examine the people, the breakthroughs and the innovation driving the drug discovery and development sector. I'm Lou Rahman, Publishing Director at Drug Discovery World. Drug Discovery World is the multi-platform global voice for the drug discovery and development community. From research stage to clinical trials, the brand covers the latest innovation and expertise in the sector. Above all, DDW readers learn how to turn science into business. Today's guest is Dr. Yuning Chen, PhD, Senior R&D Manager, Sino Biological, and we'll be discussing recombinant DNA technology and its impact on modern drug discovery and drug development. First, a little bit about Dr. Chen. Dr. Chen received his PhD in protein biochemistry from Ohio University in 2012 and completed his postdoctoral studies in protein biochemistry and biophysics from Pennsylvania State University in 2016. Dr. Chen is highly skilled in recombinant protein expression using multiple host systems, protein purification, and biochemical characterization. He currently holds the position of Senior R&D Manager at Sino Biological and is responsible for the design of novel recombinant protein products and platform optimization. Welcome to the podcast, Dr. Chen.
Thank you for the great introduction.
You're welcome. So to begin with, could you just talk us through recombinant DNA technology, its origins, its importance, and its possible applications in drug discovery, and also what opportunities it creates for drug discovery research?
Of course, I'd be glad to. So recombinant DNA technology, so basically what happens is you mix the genetic material of two species together on a broad term. So actually, I think we have been using this technology for thousands of years. If you imagine the the optimization of crops and crossbreeding of animals and so on. But the recombinant DNA technology really, I think, put itself on stage, I think, in the 1970s, where scientists first extracted the gene and put it into a bacteria. So this gene can be amplified in bacteria, and it creates the protein that is encoded by this gene. So this is where I think everything gets started in the 1970s, and I think this technology has a history of about like 50 years. Until this day, it has come a long way. So besides the bacteria system that was first used for recombinant DNA technology, and now we have multiple host systems available. And besides that, once the proteins or antibodies are made in those systems, we have a variety of tools to extract them. And these biomicromolecules has becoming more and more important. They're essentially in like every aspect of people's lives, and they can be material for industrial catalysts, for instance, like most of the enzymes, as well as they can be used as therapeutic reagents for instance, like those like monoclonal antibodies that people have been talking about and have been gaining a lot of attention. And in terms of the application or the potential for the recombinant DNA technology in terms of the drug discovery, so first of all, this technology will essentially supply us with a unlimited source of proteins. And since the proteins are the fundamental executor of biological functions, they are by nature the targets of various drug discovery campaigns. So with the proteins supplied by this technology, we will be able to have a lot of material to work with and to identify therapeutic agents based on these proteins. And also in the meantime, these proteins, or especially antibodies themselves, are considered as therapeutic reagents. So they can be generated and developed into drugs. That's the two aspects I think the recombinant DNA technology contributes to the field of drug discovery.
Okay, thank you, Yuning. That was a really interesting answer. Thank you. So this is a field in which Sino Biological has quite a lot of expertise. The company has several platforms to use recombinant DNA technology for the manufacture of proteins and antibodies to expedite the process of novel drug identification and validation, for example. Do you feel that there's a lack of awareness around recombinant technology and the platforms that are available? And in what ways can different levels of technology help increase speed of drug identification and validation?
I think, like as I mentioned earlier, this technology has been around for 50 years or so. So I think people are beginning to accept, there are drugs or drug-related substances that are made from, biological systems. However, I think especially based on recent events, there are people with hesitancy regarding therapeutics, especially drugs created from cell lines that are derived from a human resource. So I wouldn't say there's a lack of awareness, but it's still a field that is warming up to people, and people are becoming more and more aware of its existence and its importance. And I think it still takes a little bit more time for maybe like most of the people to accept drugs or vaccines that are created using this recombinant technology platform. But I do feel a great promise in this field. I think people will accept this concept and make biologics as maybe like as common as these conventional drugs where you can find in drugstore. And in terms of ways of this technology that can help to expedite drug identification and validation process, since they're using recombinant DNA technology, everything is made within the cell. We have to culture the cell, grow the cell, and essentially it takes a certain period of time in terms of its culture duration. And once the culture has been completed, we would have to extract proteins, from these cell cultures. But I think currently there are technologies or technological advances in this field, essentially, that can help people to significantly reduce the culturing duration by the development of certain advanced culture media or highly potent transfection reagents and so on. And also in terms of the protein purification, I think a lot of effort has been put into generating an automated pipeline in order for protein purification. So I think these are the methods or the future that holds the recombinant DNA technology to be more and more potent in its potential for drug discovery and validation.
Okay, thank you. So, Union, could you just explain to us then the potential of recombinant proteins and antibodies when used in drug discovery?
Yeah, I think like I mentioned earlier, proteins are they're essentially the fundamental executor of all sorts of like biological pathways or biological functions, and a lot of diseases are associated with certain mutations of a target protein or the overexpression of a target protein or the downregulation of a target protein. So proteins, I would say, they are the core in a lot of diseases, and if we can have a fundamental understanding of the structure of these proteins and how they are involved in their unique biological pathways, it will provide us with invaluable information regarding the strategies that we can use to design and to make essentially therapeutics against these proteins. So the proteins themselves can be used as targets. And again, in terms of antibodies, so antibodies are very good indicators of the presence of a certain entity, of a certain antigen that it interacts with. So antibodies are very good reagents for the detection of the antigens that they interact with. But in the meantime, they can also be used as therapeutics themselves. So for instance, these therapeutics antibodies, I think people have been heard from time to time, and especially I think in the COVID pandemic, there are like antibody cocktails that are available to treat people with maybe like severe COVID. So the antibodies themselves are drugs, but during the drug discovery process, we will be able to generate a library of antibodies, I think in the range of maybe hundreds to even thousands. So how to fish out, the one or the few good candidates that have the potential to be developed into a therapeutic antibody drug is both challenging and exciting, I think, in this field. So that's what antibodies are for. They can either be the detector or they can be used as therapeutics themselves.
Okay, great answer, thank you. So when looking to create a protein or antibody using recombinant technology, what would the workflow of this process look like? And what would you recommend a drug discovery researcher doing to make that process more successful and increase efficiency?
It's an interesting question. So basically, let's first take a look at the process in terms of using this recombinant DNA technology or recombinant protein technology. So basically what happens is we need to identify a protein of interest. So let's take the example of expressing a recombinant protein. And the antibodies, they essentially follow very similar workflow. So once we identify the sequence of a protein, we then take the sequence and convert it into a nucleic acid sequence, which is the gene that encodes this protein, and we clone this gene into a entity, into a molecule called a expression vector. And these vectors are normally in the shape of plasmids. These are circular DNAs that are usually found in bacteria. And once the gene has been put into the expression vector, and we take this vector and transfer it into a host cell. So the cell, nowadays we can use either the bacteria culture for protein expression, for instance, E. coli, or there are other host cells available. For instance, people have developed a lot of eukaryote systems for this purpose. We can use yeast, we can use insect cells, and we can use mammalian cells as well. So once the host has been identified, the gene is then transfected into the host, and then the host will start to do its magic. We culture the host into environments that suits its growth and protein production. So we essentially harness the mechanism of the host cell to create the protein of interest. And once the culture reaches the end of its duration, we would need to extract the protein, usually using chromatography methods. Some of the proteins, and for instance, antibodies and other, secreted proteins, they can be secreted from the inside of the cell into the cell culture media. So for these proteins, we can just directly harvest the media and extract the protein of interest or the antibody of interest from this liquid using chromatography methods. There are also other proteins that reside inside the cell. So for these proteins, we need to collect the cell, break them down so that the proteins are solubilized into the buffer that we use to extract these proteins, and then we can apply, chromatography methods for their purification. And I think we do have a lot of experience in terms of the expression and purification of different proteins. And during the decade of operation, we do, I think, have some experience that we would like to share with the audience here. So in terms of a strategy to obtain a protein that fits the purpose for the application of a researcher, I think the most important thing is to understand the nature of the protein, where it's resided, and with whom it's interacting with, as well as to identify certain sequence features that might or might not be problematic in terms of protein and protein expression. So once we have sufficient information regarding the biology of the target protein, then we should be able to design strategies to express the full length of the protein, or if we're interested in a certain region of a protein, but there are other parts of the protein that might be problematic in terms of its expression, then we might be able to consider to remove these parts that might cause problem, but only retain domain or the motif that we're most interested in. I think there are a lot of strategies involved in terms of how to design the most suitable construct, but everything is based on a understanding of the biological function of the protein so that we're interested in expressing.
Okay, so what would you say are the key challenges in this process, and how could they be overcome?
I think protein comes in different shapes and forms. Some proteins can be highly susceptible to be degraded by host cell proteases, or some proteins can have structural features, like I mentioned earlier, that could be problematic for their expression. So for instance, some proteins will have highly disordered region within the sequence of the protein that might result in a misfolded protein, or the protein itself might contain some hydrophobic regions within its sequence that once overexpressed or expressed at a large quantity, it will form associations with itself or with other proteins within the cellular matrix and form precipitates and so on. And also, once the protein is expressed, there are points that we need to pay attention in terms of its purification, because sometimes I think the conditions that we use to, maybe break the cells, or the buffer that we use to elute these proteins, they can also be problematic sometimes to, to the stability of the proteins. And I think the strategies to overcome these challenges vary. So first of all, we would need, I think the host cells is very important to obtain a correctly processed protein, if you will. And also we would have to be, really careful and to tailor a specific, purification strategy for the extraction of the protein to use the most suitable buffers, as well as considering, adding some additives essentially to facilitate the stability of the protein.
Okay, Yuning. So host systems appear to play a vital role in the process of recombinant expression. Could you go into a little bit more detail about this?
Yes, every host cell, so every protein comes in different shapes and forms. And in order to, obtain the protein with I think the most quote unquote natural configuration, we would need to use, host cells that can impose these correct modifications to a protein. So I think with the 50 years of development, we began with these bacterial culture systems where once the protein is made, it is made without any type of modifications. A lot of proteins bear glycans, or they have complicated structures formed by intracellular or intra-protein or interchain or interchain disulfide bonds. But with E. coli or other bacteria systems, these features of the protein might not present in the protein that is made. But with the development of the field, people are starting to use these eukaryote systems. So for instance, like I mentioned earlier, yeast, insects, and mammalian cells. And these cells are more and more powerful in terms of the post-translational modifications. So for instance, to add a glycan to a protein where it is needed, add a phosphor group like phosphorylate to the protein where it is necessary, and also fold these proteins correctly, when the conformation of the protein requires anticipation of, various disulfide bonds. So basically, we're having the mammalian cells can help us make proteins that are, especially for these human proteins, mammalian cells can make them in the shape and form that is relative as close to natural as possible. And to select the most suitable host system is also very important in terms of how to process, how to produce a protein. If the post-translational modifications of a protein is less relevant, but the quantity of the protein is of key interest, then we might consider to use a system such as prokaryote systems, such as E. coli, or maybe yeast for that matter. But if the post-translational modifications is key to the downstream analysis or studies, then we would need to select the system, for instance, insect cells or mammalian cells that can produce the protein as similar to its natural counterpart as possible.
Okay, thank you for that. As we highlighted, this is an area of expertise at Sino Biological. What platforms do you offer for recombinant protein and recombinant antibody production? And what considerations should somebody bear in mind when purchasing this technology in relation to application, cost, and ROI, for example?
So we have been in this business for almost a decade now, maybe longer than a decade. So we have established several recombinant protein expression platforms. We use these common host systems, for instance, E. coli, yeast, insect, and mammalian cells, including the HEC293, which is a cell line derived from a human source, as well as CHO, which is a cell line derived from a hamster. Yeah, we cover the production of enzymes to antibodies, and also these intracellular proteins. So we have a very extensive product pipeline with over 6,000 proteins that covers anything from, I think, therapeutic targets as all the way to I think these virus proteins. Especially, I think we have extensive library of proteins derived from SARS-CoV-2 and also other coronaviruses as well. So we also provide contracted research services using our platforms to create recombinant proteins. So if you would like us or somebody else who provides this service to help you with your recombinant protein or recombinant antibody expression projects, I think one thing to keep in mind is that there are different ways or different strategies that might be required to produce a recombinant protein successfully. So even though this technology has been around and we've made literally thousands of proteins, we can still consider it, as most of our competitors or other people in this field, we still consider the protein expression as a trial and error process. So I would say to maybe expect some drawbacks. It's a new protein that need to be expressed. And also, I think this is, of course, a cost-related issue, and based on the application of your protein, we can, select different host systems, which will definitely, impact the cost of the service that we provide or other people provide. But the key question is, what would you like to use the targeted protein for? So I think that would determine what kind of route that we would need to take, what kind of host system that we need to use, and essentially the lead time and the cost of the whole project.
Okay, brilliant. So do you think, Yuning, that we are closing in on a fully automated pipeline for recombinant protein expression?
I think people are certainly making tremendous amount of efforts in this regard. We are also making some efforts to achieve a semi-automated pipeline for recombinant antibody production, essentially. So basically, we're having an automated pipeline for gene synthesis to, to vector construction. And we're having a semi-automated pipeline for antibody purification. However, the cell culture process is still heavily manual, if you will. And I think that would be something that's for the near future as well, because cells require constant attention in order to make these cells grow or maintain them at optimal level for any type of protein and antibody expression projects. And I think that being said, I think people are making progress to create cell lines with exceptional protein expression capabilities, so that instead of conventional, for instance, shaker flasks or even bioreactors, we can start to produce proteins in the form of maybe like a 24-well plate or 16-well plate. Then these will be much more, friendly, if you will, for the conversion of automations. So yeah, I think I'm hopeful that we will be able to achieve fully automation in recombinant protein expression or recombinant antibody expression. But I think there's still some way to go before we can get there.
Okay, and then finally, Yuning, what do you think the future holds for this area of drug discovery?
Well, I still have very high hope that the proteins that are generated using any type of recombinant and expression platform will bring us novel drug candidates, as well as novel formats of drugs. So for instance, I think nowadays people are creating antibodies or protein binders with multiple valencies to recognize different targets at the same time. And also in the meantime, with the development of this field, for some people are trying to ditch the cells altogether and to try to use cell extracts as this sort of like cell-free protein or antibody expression system, so we can achieve the expression of a protein or an antibody in a matter of hours instead of days. Once these technologies has grown to its maturity, it will definitely expedite any type of drug discovery adventure using the recombinant DNA technology. So I think the future is bright, but we will still have to work very hard in order to perfect our skills and make this technology more sustainable and also more efficient, so that we will be able to bring more and more potent drugs or vaccines or other entities to create essentially a better tomorrow for mankind, I think.
Thank you for that, Yuning. That was really informative, and we appreciate you taking the time to talk to DDW today. Thanks again.
You're welcome. Thank you.
More online drug discovery and development content by DDW can be found by visiting our website. You can now view it online on your mobile or tablet, or via the new DDW app, which can be downloaded via the App Store or Google Play. Also, don't forget to subscribe to our weekly newsletter, which brings you the week's latest news, intelligence, and exclusive content straight to your inbox. Thank you for listening.